What's awesome is everyone's participation today. It's some great chats and great questions. I'm assuming the other breakout rooms went well. So I don't know. I mean, right now we got about 18 minutes, so we could do just open floor, open mic, unless I don't know, Imogen, Jan, Charlotte, if you guys had anything to summarize your group discussions, or we just want to leave it open for additional questions on anything.
I just wanted to mention that I'm really excited to, meet everybody and talk to everyone, and that, that I think we should just keep all these things going, discussions going, and everybody stay in touch. And, if there's ways we can, work together or help each other out, I'm very happy to do it. So, I think it's really nice to participate in this, and I think we should try to keep the momentum going.
Yeah, I agree. I think one of the ideas is what we have the Google group, but also to turn this into a more formal Snow Leopard Network sort of group within there, and so we can work with Justine and Rocky on how to build that out. But certainly, all of you that have been participating in this module will help form the core of that, and help. Certainly, we're all resources for you to contact at any point in time when you have questions. But also, hopefully, we can continue, like Jan says, building the momentum, keep the momentum going through SLN and building this sort of genetics group.
I know one of the things that came up in my group was access to resources to actually do the genetics work, and how that varies so much across, not even just within countries, but across countries. And so mechanisms of being able to work together or get DNA to certain people, and how that all works, I think, is of concern.
Right.
Yeah, that's definitely can be challenging, and I think every situation is almost like a little bit different, and because it kind of depends on country, and then the labs available there, and there's also various other concerns on a particular project. So, I think if you guys have any issues or questions, definitely, we can help try to guide you or, provide advice. But it is challenging. I think the direction that, myself and a lot of people are trying to go is, like, to me, I see critical. The most important thing is to help develop, capacity and ability for y'all to do this work in your country, right? And that's like where we're trying to get this, where that you guys have the resources in your countries to do that stuff. And places like China, it's more feasible to do it. Other areas like Nepal or Bhutan, or it can be more challenging. And so, so I think just by you guys joining this workshop, and trying get training and working on other projects, it's essential to try to build the resources there.
Yeah, I would say one of the questions that came up in both of my sessions for the landscape connectivity is obviously like the analyses portion, right? So like, we spent a lot of time talking about how to collect the data and what we can do with the data. And then obviously like looking at the wet lab work, but then obviously once you have your raw data, what exactly do you do with it? And of course there are a whole myriad of there's a whole suite of options, but then there's also a whole suite of opinions on how we can do that. And so, realistically, I think that for many of us, when we are getting to that point, it's going to be really important to be able to have this type of network to work with, because there are so many different, just in the spatial context alone, there's so many different analyses that we can do. And it's not necessarily about determining which one is best, but it's about determining which one is the best for the data that you have and the question that you have. And so being able to both have someone, a network to bounce those ideas off of is important, but also even comparing data sets and knowledge bases can be particularly helpful. And so, I would like to see a continuation of that for all of us here when it comes to actually analyzing that data, because the reality is that everyone here is going to have, different pieces of knowledge that will be helpful for different projects, whether it's combining data or also just sharing different packages or data analyses or even tutorials on something. And I think that would be really important for all of us going forward.
Yeah, so Earl Shamus mentioned in the chat here that this all sounds great, and that it's mostly due to lack of guidance that research projects like these might fail, where those very minute details matter, and just not having the wherewithal. No one tells you that it's really important that you wear gloves when you handle different samples, or all those different these little details that can make a big difference. Like that contributes to some of the challenges. So having this venue, I think, hopefully is going to be helpful to Erwan and others, but also understand it doesn't stop today. That this, again, we want to build this group and be a resource. And yeah, the end goal is that ultimately there's capacity built within countries by the people of those countries to move this work forward. And our role, the four of us sitting here in the United States is to help facilitate transfer of knowledge and guidance and those sorts of things, and connecting different groups to one another. So I think we're all open to helping in any sort of way like that.
Yeah, and I would also, just kind of following that up, is that it takes a lot of time to be able to do this well. So just you're gonna get frustrated, you're gonna struggle, problems of contamination, and there's a pretty big, we kind of make it sound like, oh, it's pretty simple, you can generate the genotype, but there's like a pretty big learning curve with it, right? And if you it really does take years to kind of like get really good at some of this, where you're learn it enough where you're confident, and when you have bad data, okay, it's the samples, or is it did your reagent and one of your reagents go bad in your kit, or is it because it's contaminated from the field, or is it because the samples degraded, or was there a did somebody switch the samples when they were loading it on an instrument? It takes a lot of experience to sometimes get good data and analyze it. And the other thing is, I sometimes see comments that microsatellites aren't good for a certain thing, or just microsatellites are bad or something, or just like non-invasive genetics doesn't work, or it's like anything, right? There's a lot of ways you can screw it up, and you need to be able to do it right. And there's resources to help you do that. So I just want to kind of highlight the fact that it does take experience, and you can get that experience, and you can even learn it on your own if you're motivated, right? But you do need support, and you do need help, and you need assistance, and it just takes time to learn how to do. Like when I was starting out in my master's, right, I wanted to do bobcat genetics using non-invasive scat samples, and I learned how to do genetics on scats, and I completely failed at it. And I couldn't get the scat to work, and I ended up using blood samples from the researcher who was working in the same area, and he had radio-collared bobcats, right? And so like trying to learn to do these things with scat is actually not a good thing to learn on. Like it's best to learn extraction and microsatellite genotyping with like good quality tissue samples, and blood samples. And if you're in a lab like overseas, and obviously you can't get blood and tissue from snow leopards, but you could consider trying to get samples from, let's say, maybe domestic cats, or get a zoo sample or something that you can learn the methods on. And when you can do it well, then kind of like switch to doing it with scat. That's another thing I would like recommend is learn with good samples first, and then apply it to the non-invasive genetics.
Yeah, doing genetic work on non-invasive samples is like starting at expert level. And if you haven't ever done it before, and you're like just a novice, it's really hard to make that jump. So yeah, that's a great point on that. If this is a way you see yourself moving forward, or want to work on these projects, like go and volunteer in a lab and just spend some time getting the basic lab skills down on like an answer is good, good quality. Set yourself up for success, for some wins, build some confidence and then go to expert level. Does anyone have any other general questions or comments? It's sort of your chance to, Imogen mentioned, the need that, I think we said at the very outset that, these 4 weeks of this module were going to be more, not so that at the end of it, you're going to be ready to off and run and do a genetics project, but that you would have some of the background understanding and terminology and sort of foundational basics of what it would take to start asking the questions and how to do a project together. So if there were to be another module or another mini module, or is there any particular areas of topic you'd like to see focused in on or specialized or more time?
I think that was pretty, thank you all for like the very nice module, but I think that was already pretty good for the ones that were not introduced to genetics before. Like it really gave like a spectrum, and for that, like for now, it's hard, at least for me, to think about what more. I think the collaborative work, how to implement that really, what if we don't have the infrastructure was the main issue, but that's more logistics than science really, so. No, it was great. I'm gonna miss this.
Thanks, Sherry.
Yeah, it's true. It comes down to logistics. It's like, one thing to like you can do this, theoretically, or it's possible. The technology is there, but logistically it can be difficult. And I think maybe we could discuss with SLN, like trying to put together just a list of potential labs in different countries that might be suitable for analysis, or be willing to do genetic analysis on snow leopards, and maybe like that they would be like a contact point for sharing resources, or or where to focus some of the follow-up training or something, because I know like there's also different levels. Like some of the labs, like some of the labs can do DNA extraction pretty easily. Some can do up to PCR. Other places could sequence, and some might even have access to like an Illumina instrument. And so so that would seem like that would be very helpful for people, and maybe like through the SLN we could try to put together like a list of contacts in labs.
Mm-hmm.
Yeah, it's a great point. So, for example, I guess it was last year and a half ago, Imogen and I did a workshop in Tashkent, Uzbekistan, where it was at a lab. It's through UNDP was supporting it, but there was a lab there, amazing state-of-the-art facilities. It was just focused on cotton genomics. So it was just a lab that was working on something completely outside of sort of, what we're interested in doing here, but they just wanted to have that training to say, all right, how do we shift that knowledge base and these facilities to use more towards this non-invasive genetics? And so we're instructing and working with a group of really experienced lab techs and people. It was just a matter of not starting from the basics and moving up, and saying, how do you shift those skill sets to work here? What are the same, what might be different? And so, I can imagine across many of these other range countries, maybe similar labs existing and not needing to be built from scratch, but more, how can we go in and adjust and tweak some things to make them.
True. Yeah. There's a lot of this technology, it's they kind of like wildlife is the last place this stuff gets integrated, right? So like, in like Pakistan, and India of course has a lot of wildlife labs, but like in Pakistan, maybe Kyrgyzstan, Nepal to some extent, like these are the labs that have these instruments probably focus on either agriculture or human health, right? And so, but there's still people there that are interested in, potentially expanding to wildlife. So yes, I think that's important to be able to identify, potential labs that have the instrumentation technology that would be open to like expanding into wildlife. Yeah, like Byron has a great example of that. So I'm guessing Mongolia is probably also similar in that. Like I think I was there. It's been too long since I've been back, but I think the last time was 2012 maybe, and we had visited a lab in UB that I think was also maybe a planned agriculture plant lab, and they and I think there's also a veterinary facility that I think we visited, and they're that typically like agriculture, they just have more funding than wildlife in most places. So, and then of course the human health stuff.
So Helena wrote here, it would be great to know what labs may be open to us getting involved for more experience related to snow leopard genetics or even big cat more generally. And I would even say, Helena, if the idea here with your comment is just to get more hands-on experience, but this, in many ways, the species doesn't matter. Just understanding how to work with DNA from whatever species and building up those wet lab skills will be universally applicable no matter what you're working on. Of course, it's more interesting if it's cats. But yeah, so I don't know as far as Jan or Charlotte, or even if you guys have thoughts on, like, I don't know, Helen, are you thinking like with regards to like internships or volunteer opportunities to get into a lab, something like that?
So I mean, so at a university, like usually, like in my lab, for example, like we bring in undergraduates, right? But typically, the people who like end up getting experience, like in my lab or the labs in the biology department, they're they're pretty much the undergraduate students there. So, like if you're at a local university or you work near a university and you're not a student, it can be harder to try to like get experience for sure. I mean, if you're a student, then it's pretty easy. It's just a matter of finding a professor that does genetics and just harassing them till they let you in the lab. But if I get I know it's really challenging for people who are not in a university setting, right, or not students anymore. So that's definitely even challenging. Like I've had a couple of times where I hosted researchers, from abroad. And Charlotte worked with one, Katie Sipra. I don't know if you guys have met her, from Pakistan. She visited us for a week and a half or 2 weeks. It was something like that. And, we kind of ran through the extraction and genetic analysis. So like if you're not, if you're not in a university setting, I think the best bet is to try to, contact some lab where you could potentially visit for like a week or two and maybe, get trained. But it's definitely hard to find opportunities where you can like, volunteer, for a longer extended period of time, right? And then of course, if you're gonna get trained, like if you are gonna go visit a lab, you typically have to come up with the money yourself to travel there and all that. And, I mean, I think there's a potential, maybe there's like, smaller grants that you could apply for to do that, but it's a problem. And I mean, I think the suggestion that Byron makes is the best is probably like, don't limit yourself to only labs that do snow leopard or carnivore stuff, right? Basically try to find a lab where you live, where you can maybe go in and volunteer or spend some time there. And, as long as you're learning molecular biology, you're going to learn the skills to then, transfer that to genetics. So, yeah, so it can be challenging, and I think we've always talked about potentially doing it, workshops around the, different places in the range countries. But again, those are usually pretty limited, right? And that's the problem is like, to really learn it, you need to be consistently, doing this stuff. So I think your best bet is, to try find a lab locally where you could spend some time in.
Yeah. Imogene, so I guess we're out of time now, but I don't know if Imogene or Charlotte had something you're trying to jump in to say or comment on, or otherwise, we're getting a wrap up.
Yeah, I'm good. But yeah, it's been a fun 4 weeks. Thanks everyone for joining. It's been really nice to meet you all and chat with you and get to know you a little bit better and be able to build a platform to spread genetics a little bit further in snow leopard world.
Yeah, I like the same. I don't have anything else to close out with other than I'm certainly hopeful that we can remain in contact. Anyone here is welcome to email me. And if I can't answer your question, I'll point you into the direction of someone who can. It's likely going to be one of the instructors here, but obviously there are a lot of people in the world, including people here in this group here that are attending that are also really great sources of knowledge. So I look forward to connecting.